equations and stuff. That's really works. It's called meeting owl, like a bird, owl, O-W. Yeah, so I don't know. Yeah, not far away. I think it's a 10-feet range, which is like 3-meter range in there. So for a small room, round table, it's perfect. For a speaker right there, if someone on the back, they can come closer if they want the offline people to hear it. So, yeah, no, I mean, but I mean, you guys have this system, so it works. But that's probably like 20,000 or 30,000 to install. All right, should we go ahead and get started? Okay, so again, given the small audience, I want to really keep it informal, so stop me, ask questions. I don't really have to get through everything I have on my slides. I kind of want to give you like a little bit of flavor of what we are doing, and again, I'll try to keep it sort of very non-technical on the bio side and relatively non-technical on the math side, but we can go into details in there. So I'm at Rice University. Rice doesn't come from Rice the Great. It comes from this guy, William Rice, like a rich Texas guy who like died in the turn of the 20th century and left his money to establish what was Rice Institute and now Rice University in here. And we are right there in this Bioscience Research Collaborative. So Jose Onicek, which I think some of you know, is on the 10th floor. I'm on the 7th floor. We are part of the same center and stuff. So anyway, so like kind of give you a bird's eye view of what my group is doing is we sort of tries to, I mean, I'm physicist by training, so I speak your language, but I've been in bioengineering department as a faculty for a while, so I have to convince my colleagues that what I do relates to engineering, even though I can't make much things that are working. So in a sense, I sort of redefine science as a reverse engineering, and that's really what we do in biology. We have a system that works, and we are trying to understand how it works and why that was it built by evolution the way it was. So kind of we are trying to understand some engineering type of principles there. And a lot of things that we do is sort of biochemical networks and stuff, maybe so in the intracellular scales, how feedback loops affect things. But what I'm going to be talking today is more on a multicellular scale in there. So again, to give you a little bit of an overview on what we do on intracellular scale, a lot of things we do is kind of stochastic thermodynamics on equilibrium, statistical mechanics of reactive systems. We've been sort of trying to understand biological catalysts, enzymes, how they sort of optimize competing design goals in a sense. They need to be fast, they need to be catalyzed in the thing. So think about making proteins, you insert amino acids one by one, and you do that. But on the other hand, you need to be precise. You need to make sure it's the right amino acids rather than chemically similar ones. And they also do it by burning energy in terms of chemical fuel, ATP. So how those three things save energy, be fast, and be precise, how those competing goals compete in there. So we also work a lot on how sort of cells sense things and make decisions. So for example, we've been trying to understand sort of what are the sort of feedback architectures that allow them to make decisions and go from one state to another on the dynamical network type of thing. But what I'm going to be talking about today, and it's probably best for the physics audience, so you guys choose the right one, in there is really try to understand multicellular self-organization, multicellular pattern formation. So we've been trying to understand how sort of cells, and for the most time of the talk, it will be this bacteria with a complex name Myxococcus xanthus, but I will call it Myxo. In there, how it sort of self-organized into amazing cellular patterns, like this is all examples from the papers we published over the years. So we also looked at some self-organization in other systems like fibroblasts around the tumor and stuff, but I will be talking about the mixing here. But as a motivation, I mean, it's still a fascinating problem, and it's complex and unsolved problem how an organism as complicated as we are grows from a single cell to all this complex entities, different cell types go to different tissues of the right shape, of the right size, and stuff. And in the course of what biologists call development, growing from a single cell to multicellular, there are a lot of things that are important. Cells need to grow and divide. They need to talk to one another, both through chemical sort of signaling and through contact-based signaling. They need to move. Their cell motility is very important. And they sort of needs to sort of reorganize their environment for themselves. So this is a very complex problem, even for best studied multicellular organisms, like maybe fruit fly or small worm, which just have a handful of cells. We're still not very close to understanding all the intricacies in that. So my take is that the bacterial model systems, doing that in bacteria is simpler, and we are actually close to figuring out a lot of complexities, and we can learn some general principles in here. So all those processes are also happening in bacterial multicellular communities. And this is one of my favorite bacteria. I work on many bacteria, but this is one of my favorite. It's called Myxa, Coarse in there. So, I mean, from the point of view, it's a long rod, about 1 to 10 aspect ratio in here. It's flexible. I'll show you some pictures on the next slide. I mean, nothing special. There's tons of rod-shaped bacteria over there. It moves on the surfaces. It doesn't swim in 3 dimensions like some other cells, but it likes to crawl on the surfaces, and it does it about 1 body length per minute. It has 2 engines. They are different biophysics and different roles in there called A and S. I'm not going to go into details. Engines, some engines are sort of polar. For example, the S engine has to do with the filaments that are secreted on the other side. So it's kind of like your efficient rod. You hook on something and you pull yourself there. And of course, you need to do it from the front end to go there. So cells periodically, every 5 to 10 minutes, it disassembles engine on one end and reassembles on the other end, and cells start going in the other direction. So it kind of makes it goes back, but not like a car which makes a U-turn, but like a train where conductor goes and goes to the back of the car in there. So and the synchronization of those changes of direction, that's really important for that group behavior in there. So and there are chemical signals that can affect how often you do that switching, and there are also contact-dependent signals that do it. So and when you talked about, say, you guys had evolutionary ecology, you think what defines an evolution, from evolutionary biology, what defines an organism to be a multicellular? There's a checklist of things that they do, and of course, we as multicellular eukaryote and all the animals, we have all those different cell types. But if you look at some single-cell organisms and mix and some with close relative are in that category, it actually fits all the checkboxes. It can be defined as independent evolution multicellularity. Social amoeba-dicty is another one that also checks for it. So even though you think about it as a single cell, it actually have a very sort of interesting multicellular lifestyle in a sense, because it's, and the reason it does it is also very interesting. It's a predator. It eats other cells. And it can eat other bacteria. It actually can eat other eukaryotic cells, some fungi and stuff. It can eat other things. But then you imagine, how can a small thing eat a bigger thing than it is? It needs to digest its foods outside. And basically what happens is collectively they go around their prey, they put something to kill them, and they put something like digestive enzymes to do it. So their digestive system is outside of the cells, and they just take in there. And of course, when that's your lifestyle, you better be social. You wouldn't survive on its own. I mean, on a lab, you give it the right nutrients, you can grow it from a single cell to a colony. But in the wild, they need to be together to survive. So that's sort of the kind of the biological story of what happens. So anyway, and because of that, it's really was discovered. When people discovered it like 100 or so years ago, first you can see some of the structures it forms with naked eye. They can see millimeter size, like mushroom type objects, and that's Myxo does it. So people thought that's a fungi or maybe some sort of yeah, but then they discovered that's a bacteria, and since then, or maybe not since then, but maybe since the 60s or 70s, it was studied as a model organism for collective behavior. So that's why we know so much about it. So even though it's small community. So again, this is how a single cell moved. This is how a single cell, and this is not a movie going back and forth. This is like the real reversals in here. But indeed, what we are really interested in sort of pattern formation, how millions or hundreds of thousands of those cells together self-organize into different structures. So when it spreads out on the surfaces, it forms like fractal-like patterns. Yes, they move a body length percentage. I think it's about 100 times speed up or something like that. So probably, yeah, something like that. So this is time in minutes. So 2 hours, 3 hours in stuff. So I don't have the time here in there. But like if you see here, a single dot is a single cell, and you can see it goes back and forth, and the period is 10 minutes, and it's probably 10 seconds here, so about 60-fold in there. So yeah, anyway, so you can see that at some conditions they self-organize and travel in waves in there, and they do mainly that during the predation. When they cover their prey, they self-organize and travel in waves. This is actually a phenomenon that got me interested in this bug and got me interested in biology to stay. I was a PhD student doing a rotation in a lab of George Oster, who was working a lot on molecular motors. And there was a postdoc in the group who was trying to find biophysics of how those cells produce force and form. And I started reading about that, and I see those patterns, and I blown away. I said, I want to work on pattern formation, and I got in there. Yeah. So yeah, so basically in there. But today I'll mainly talk about how they self-organize into those mounts. So this is a electron micrograph, but this is a time course. Again, time is in hours. And you can see that starting from uniform distribution, they come and self-organize into those mounts. Some mounts will disperse, some mounts will merge, but eventually you get those hinge mounts. So we are trying to understand basically how do they do that. And I've been sort of doing it for quite a number of years now, and we have a lot of sort of different tools in our toolboxes. And I will kind of go through a few of them today, sort of showing you how we're gonna do it. And the first one is maybe somewhat unusual for this field but I feel like it's really powerful. And again, one of the general principles that we learn, and I think this is general principles that's definitely applied to other biological systems as well there. Biology doesn't like to work as would be exciting and interesting for me as a physicist on a toy model basis, where you have like a one leading term of interaction, and that's what drives the instability in there. What happens there is that you have like sort of multiple alternative toy models, so multiple alternative things that could work on their own, but what happens is this collection of those interactions really does it. And I was thinking a lot why that happens, that I think it's for robustness, because different sort of instabilities should work maybe in different environments, and what happens, this partial redundancy and synergy of those instabilities is what drives it. So we've been really trying to sort of learn from data, and this is again beauty of the system, is because unlike other sort of like human development or even like say fruit fly development, we can get a lot of data quite easily here. And so we've been so what I'll talk about in the first part is how cells self-organize into aggregates, and what are the cues or what are the sort of cell behaviors that drive in there. So this is something that sort of we published a while back, and we build on this method in a subsequent publication, but this is the idea. So the idea is, let's collect lots of data of how cells move. Let's sort of, and then learn from this data and figure out what are the intercellular interactions that could be responsible for this aggregation here. So, and in terms of the data, again, I don't have a lab. I collaborate with people in there. But when the typical things that you get is something like that. It's a sort of electron micrographer, and this is the images you have on the previous slides. It's usually like in a phase contrast microscopy or Rugel bright field. And you can see some aggregates start forming here, but it's really no one-to-one correspondence of this gray scale intensity and the cell density. So that's sort of a problem for us. We actually published recently a paper that takes those images and trained deep learning networks to convert them to effective cell density or fluorescent microscopy images. But for this type of things, we really did it with fluorescent microscopy. So what you do, you take a cell and you make it produce a fluorescent protein. And we actually have two colors of fluorescent proteins. One is sort of relatively bright here. Another on that wavelength is just a little bit of a sort of tail of the emission spectra. So outside of them, there's all still plenty of cells, and we can get a cell density from there. So this is what we pretty much get. We as time goes in, we have 0.1% of cells that we can track, and we can easily segment them, and we can see how they move as a function of time. But at the same time, we can filter out those cells and take and smooth the image what's below them, and that's the cell density. So we can look at cell density as a function of time, and as the aggregates will appear, we can really segment them as a simple density threshold. This is what happens in there, and this is the segmented images, this is the raw data, and you can see that we can sort of track how cell density changes as a function of space of time and how cells move as a function of space of time. So this is a rich data set, and now let's try to reverse engineer what individual cells are doing to produce that type of thing. And pretty much that's how we have cell behaviors and we have cell densities. And in this cell density, because again, cells are talking to one another, or cells modify the environment and change the environment. So cell density might be related to behavioral cues. So of course, this data set is rich, but we need some sort of coarse-grained version of that. So we want to sort of take those cell trajectories and divide them into segments. And the way the mixer moves, as I told you, it goes back and forth. So we want to see how often, and this reversal frequency is important. It changes as cells get in there. So we try to figure out, and also in those things, it also sometimes stops, or it just starts jiggling around because other cells bump into in there. So what we do, we track cells as a function of time, and we detect the reversals, so green to there, and then you'll have some yellow segments where cells kind of stop and stuck in place. Here was a little bit of that in there. So you see here cells actually make a U-turn here. Here it's sort of regular, and we kind of can do it with a simple like Kalman filter-like thing. Let's detect those three types of behavior, and we sort of discretize the trajectory in there and then put them in one big database of cell behavior. And so let me just go past that in there. And one thing that we can now quantify is what's different, say, inside the aggregates, near the aggregates, away from aggregates in there, or what's different as a function of cell density. And like one of the leading models of aggregation at that point was kind of a traffic jam model of aggregation, and you guys in São Paulo probably are very proficient in this type of model. So when we have more than a critical density of cars on the road, they can move as fast and they slow down. As you slow down, the density increases. You get a further slowdown type of thing there. So, and it's a negative correlation between speed and density, and that's actually what we see. But in and out of the aggregate, you can see that inside of the aggregates, the speed is slightly slower than below the aggregates in here. Blue is this state in which cells sort of persistently move. The red is this non-persistent. This is kind of a stop state, but they still move a little bit in here. And you can see that basically they move for shorter, inside of the aggregate, they move for shorter time and with small speed. So you get about 40% of difference in the free around length in here. So that's what we see. So, and then the other thing is that when they end up their run, and they either can go back or they can stop and stay in position and be pushed to swing. They have about twofold decrease in probability of continue moving the right way. So they slow down their motion and they're more likely to stop. So, and if you sort of combine this and just say what would be the run length as a function of cell density, as you can see that it's about 2.5-fold decrease at high density inside of aggregates than in outside aggregates. So this traffic jam effect is very definitely there. And if you do, since we have a lot of statistics, we can have a very good confidence of the mean of that. So you can see a lot of things there. But if you look at the spread of the data in each bin, the noise is enormous. And you can kind of see it here. You can see this is the range of the data in here. So what you see, we have a signal, but we also have a very high noise. So the question is signal strong enough to overcome the noise? So for that, we use what we call the sort of data-driven models type of thing, in which is sort of, I would say it's kind of a non-parametric model in which everything is in the data. All we put in is what things is correlated to one another. So what we basically do, we start with sort of agents in the simulations that are sort of the same size as our experimental field of view, and then we sort of experimentally sort of get numbers from experimental database of behavior. How long is the run? What's the duration of a run? What's the speed of a run, which is one divided by the other? And then we move those agents and then sort of keep on going. And of course, if you start doing it, it's completely integrated. No, you just sample from all the behaviors you have. Nothing will happen, and this is what sort of we'll do. This is nothing here by noise, no aggregation, because I mean cells just do what they do. So in order to get the self-organized behavior, you need to introduce some correlations in the cell behavior. So cells should be sensing something. And if you want to, for example, look at this traffic jam effect, we want to make the cell speed or run speed and probability of going backwards or stopping be a function of cell density. So when we sample from that, we sample conditional on the cell density. So it's instead of just sample from it, you sample, I mean, and there are multiple ways we can do it, and it doesn't matter. So this is what we do. So we basically, and this is an example of an open loop model in which what happens inside of our simulation is completely driven by the experimental data. So instead of cells sensing their own density, they're sensing the density of experimental cells in that location. So in this case, when you run it, you should be able to reproduce the aggregate exactly where they are, because and you should see the same ratio of cells inside the aggregates and outside of them. And basically we do this, and I'm not going to go through the algorithm, I just play that, and this is the model played on top of experimental movies. And at some point you can maybe notice there's some clouds of red points, they come and it's in the white region. So indeed there's a little bit of aggregation of cells inside of it. So traffic jam models works. I mean, traffic jam, and you do get instability. You don't have a uniform distribution anymore, but it's still plenty of cells outside of the aggregates. So it's not all the cells, that works, but it's not, the signal to noise ratio is not enough to cause all the cells to come to the aggregation. And so, like, so what the movie played, this is minutes. So overall, I think 6 hours comes from the initiation to the aggregation. The whole process takes like 24 to 36 hours, but after the mounds are formed, cells inside of them will start form spores, and there are also biochemistry going on, but the motility problem. When they're starving, they don't. Normally, they divide once in 12 to 24 hours. So 12 is the minimum. But at that time, you have, when they're starving, they know they're starving, and they change their behavior. Now, instead of spreading, they stay in place. So instead of sort of chemataxin out, they stay in place, and they start aggregation, and they stop division at that point. So in those, I think in all I've been showing you, cell division is not important. Mm-hmm. Yeah, so that's a really good question. I think it is. And part of the reason we see slowdown, because I think when they do, they move in there. But our data, our microscope is there, so we work on them in projection. There have been some beautiful kind of an active matter physics role of how the first, second layer forms, and actually it forms from defects in pneumatic crystal filled in there. So, so the second dimension is there, but at some point they are able to go into multi-layer situation, and that's what the aggregate is. But we are looking at the projection, so some of the slowdowns is due for them to going up in there. Probably, so as I tell you, aspect ratio is about 1 to 10, and probably the typical aggregate, mature aggregate, can be 10, 20 layers easily. So it's comparable. Some cousins can form like a branch tree-like structures. I would love to have an experimentalist who studies them in the lab. Apparently they are not domesticated and they're hard to grow, so people don't do it. I would love to get my whole product. The patterns are just beautiful in there. But this one forms a boring mushroom type structures, and that's the one we study. Yeah. So again, when you put them on as a drop, they put in, the drop dries into the gel that you put them on, and then they start moving. At that point, they can move in there. But then when they spread out, it's only the bottom layer that moves. And in bottom layer, if two cells collide, they will do it. I will show you some of that in the bottom. But during the aggregation, at some point, they know that they want to start forming this, so they start crawling on top of one another. Anyway, so you can see here that there is a little bit of aggregation, but only like 20% of cells get to the aggregates, compared to something like 60 in the same time course, 5 hours in the experimental. And this is the comparison of the final patterns by the 6 hours. You can see this is the experiment. You get the same positions of the aggregates because this density was driving the behaviors, but not enough. So what I'm saying is that right now this model didn't have any free parameters other than like numerical things when we coarse grained this. We see that in this case we haven't figured out all that it matters. And what we do then, go back to the drawing board and see what else is happening. And one thing is happening, that cells that go towards those aggregates, their time of going before they switch their behavior is longer than cells that go away from the aggregates. So it's some sort of a taxis. When I go there, I go for longer, and then I turn back, and I maybe go to sooner. And that also the probability of stopping is changing and stuff. But this is like, if you look going towards and going away, there is a very significant differences, and that's not in the speed. So it's not like Oswald ripening or something like that when there's an active gradient of chemical potential of something that does it. And that was not a crazy idea. So cells produce surfactants, and those surfactants can produce forces on them and stuff. But so there's really no difference in speed. It's really sensing. It's really difference in them adjusting their behavior, adjusting how often they rebuild that engine and switch and switch it. So once you put that in, things start working, and they work almost perfectly. Maybe not quite as well, but I mean, I would be happy with this, because again, our cells move on straight lines. It's coarse grained, so maybe too much noise in the coarse graining process in there. But of course, to really test the things, we need to test it in a closed loop fashion. We need to sense the density of themselves and see sort of random self-organized pattern with random position of the aggregate, just like when you do experiments multiple times, that would be good. And that's sort of the closed loop model. The only thing that happens is that we estimate density from position of the agents, and that drives our biases in our sampling and stuff. And when we do that, what happens is it takes much longer to nucleate those aggregates. Because once the aggregates are there, then you have taxes. Before the aggregates, it's not. And pretty much this two-dimensional random search, even if you have a little bit of density, that's not enough. And that's what you see. That eventually, if you run it longer, it will accumulate, but there's a big delay in here. So, and whenever I play the trajectory movies, you maybe see how well aligned the trajectories are. And we really, at this point, our cell angles were somewhat random. So what we were missing is actually collective cell alignment. And this is what you see. You see right here, it's trajectories over the course of 6 hours. And here they almost move unidirectional. And you can see that this unidirectional like a stream, and it points towards the aggregate. So instead of two-dimensional search for a target, you do one-dimensional, so which is much faster. So we need to account for those correlations. So now when we do our simulations, when you search for an angle, we do sort of correlations with angles of other agents in this temporal and spatial vicinity. And when you do that, everything works beautifully. We have complete agreement between how many cells get into the Freudian bodies, what would be the density of cells in the aggregate, what would be the size of the cells in the aggregate, how many aggregates will be formed. So right now, those three things sort of all matter. They get no free parameters in there. And this is what happens. So cells decrease their speed at high densities. They bias their reversal time to go longer in the direction of aggregates, and they align with neighboring cells, and there's also alignment in the sort of in the direction of aggregates. So now the question is really, so those are the behaviors, what is the mechanistic basis of this behavior? Between bias and what? Oh, so so one is like when cells start moving, it's much more likely to be in the torus or away from the aggregates rather than in the radial direction. So this is like a pneumatic type of thing. And when you move on along that thing, you move for longer here than away. So one is about orientation, the other one is about times. So both of them contribute in there. So what I'm going to be talking next in the remaining time is like what's the mechanistic basis of those type of behaviors? Because right now what I say is this is the correlations that matter. This is the behaviors that matter. But why did they matter and where did they come about? So let's me talk about first on this is sort of more on the active matter type of flavor of biophysics of cell alignment. So how the cells sort of self-organize into those aligned trajectories, where are they coming from? And I mean, physicists work on self-propelled rods for quite a while. So, I mean, that seemed to be like a sort of piece of cake when we started working on it. I mean, there's multiple things where rods colliding with one another get together and form those clusters of things. But there are two things about mixer that were not in any of the models that we've seen. First of all, we see how flexible they are. So we've been trying to see how cell flexibility matters. And there have been some work in there, but there were conflicting evidence in there. So you can see there. So this is like a flexibility in there. And then cells also periodically reverse. And people who were doing this self-propelled rods, they were mainly motivated by, say, molecular motors on the substrate and microtubules on there, and those things move unidirectionally. So reversal is very important. So we've been trying to see what will it gives in there. And what my student did, he sort of developed this sort of way of spring and nodes model of cells with multiple segments, with linear springs that keep the cell length constant, with angular spring that prefers cells to be straight but still can be flexible. We can estimate realistic flexibility of cells from other things, and then we just solve Newton's laws of motions, and of course it's over-damped limit, so there's no inertia there. If cells velocity is proportional to the force, the rotation is proportional to the torque in this limit. But this is like what happens as when two cells collide, and this is similar to the movie that I played in the previous slide in there. So but this was actually not trivial because we've been depending on how cells interact with the substrate, whether it's completely sort of viscous interaction from substrate, so it's just a friction with constant force or force proportional to the velocity, or elastic interaction with substrate. There's really some binding proteins that you need to pull. You get a different outcome of this collision. And at that point, there are two alternative biophysical models of how cell propulsion is generated, and we were able to disprove one and confirm the other in here. So for the purpose of this talk, I'm interested in what happens in the large collectives of cells, and kind of running this for thousands of cells in simulations, you can see that naturally by this pairwise of attractions, you form those clusters of cells here. So what you can notice in here, cells don't reverse. And of course, there are mutants of mixa which don't reverse, but normally cells reverse. So this is what happens for non-reversal cells. And what we see in this simulation is relatively similar and quantitatively similar if you look at cluster size distribution and other things that people quantify in there. So you get those large clusters of cells, you have moving together, you have some distribution of cluster size, and we can quantitatively match that with a few free parameters in here. So everything works. Now cells reverse. If we can just turn in the reversals, right now no taxes, nothing, just a random patient reversals, all those clusters get dispersed. And this is what we see in simulation, but this is not what people see in experiment. In experiments, cells actually form big alarming groups even with reversals. So we must be missing something. And what we're missing is what you can see in this movie right here. So I played this movie, and this blue is just a false colored track of where the cell's been. And this cell is gone, but the track remains. And there, apparently what will happen is this big group of cells will turn and follow the tracks of the cells, even the cell is not there. So there's some sort of orientation memory. Those cells, kind of similar to ants, they lay a trail, they pull electrolyte gel, and that actually allows other cells to form their stuff. So there is this orientation memory, and this orientation memory is apparently very important to get in there. And again, if you start with the reversion cells and you put this orientation memory in there, you get self-organized patterns of this sort. No, there isn't. I mean, we try to quantify it. No statistical significance. No, I don't think in there. So I think it's just like list friction in there. It also might be a grooves in the agar that they do. So there are multiple. I mean, grooves in the gel that they burr, and it's easier to move in that case. No, I don't think so. I mean, they're already on the trail, and even single cell sort of goes back and forth in there. So as they go along the same trail maintenance, it might be self-reinforcement going on, but yeah, in there. So I don't think those. But yes, basically we get this type of patterns, and you see it's kind of a network of trails where cells move, and no cells in between. And if you look at the same thing 2 hours later, pretty much white space will remain white space. So it's a different type of thing. And that's exactly what you see in the experiment with wild type normal cells, is that there's a network of things in white space. And maybe occasionally a cell will separate from the trail, goes on there. But for the most part, those trails are connectively in time. So anyway, so mechanical interaction, slime strand following, observe the patterns of what you see in low density in here. So now we want to try to go and use this model or some version of this model to try to understand alignment in a different condition where instead of 1,000 cells in a single layer, we have many more cells in there. So for that, this is a work of my other students. We really needed to go from a detailed mechanistic model of Newton's laws of action and 20 variables or so per cell to something much more phenomenological. So cells, how cell alive is the neighbors in cells. And basically what we try to understand is, I mean, cells alive is neighbor in cells, is that enough? And cells align radial to aggregate. And this is really interesting for me because we know that bacteria cannot actively steer something. If cells, for example, sense a gradient, if like big cell, like a cancer cell, it senses a gradient of something, it has receptors on one side and on the other. And those receptors trigger polymerization of internal proteins inside of the cells. And those cells can actually make a turn and go. Bacteria, if you can do the back-of-the-envelope estimation, they're too small to sense gradient directly. They cannot actively steer. So if we know that they align and go in the radial direction, that might be emergent phenomenon. It's not on the single cell level. It's something on collective here. So we are trying to understand how that is happening. And pretty much the hypothesis that we have, and again, this is probably works as well for St. Paul as for Houston. If you get rid of the city, but just leave the road maps of Houston, the freeways and stuff, you will pretty much figure out where the center of Houston is, because all those freeways that go from the suburbs to the center, that's all the thing. So that's what we think is happening, is that when cells self-organize into those trails of things, most likely the aggregation center will emerge where those paths or where those things interact. So that was our idea. So we try to understand how it's happening. So we need to speed up the things about 2 orders of magnitude. So we need some coarse grain approaches in which not every single cell is simulated. And so instead of mechanical, we need some sort of coarse grain. And yeah, so this is how bacteria usually grow, called agar. So you put some nutrients and you put something that jellifies that, and you pour it, you make it slick, and now inside of that there's nutrients that every cell moves, and then you put a drop of liquid in there, they suck into that, and they start moving on top of it. Yeah, of course. If you put plenty of nutrients that they like to eat amino acids. If they put plenty of them, instead of aggregating, they will be spreading out. If you put, for example, certain chemicals that makes them reverse like crazy. Mm-hmm. Yeah, so for example, you can make hard agar, which is like low liquid, and low, and yes, you can, and basically, I told you they have 2 engines, and 2 of those engines work differently well in hard versus low agar thing in there. So this is, I mean, I'm kind of talking about both of them there. For example, on glass with some covering, they can also move. And there, they mainly move with the one type of agent. You can also make like some sort of very sort of soft type of environment when they burn holes, and there they mainly pull themselves out. So yeah, there's some interesting biophysics there for sure. So pretty much what we do some sort of phenomenological type of model of alignment, and this sort of alignment is sort of pneumatic alignment, which is sort of like Vishik-like model. It's, I think how many people are familiar, but pretty much it's one of the sort of cornerstone of active matter papers when people say that in this 2D active matter you can have phase transition in the Ising-like model when particles are moving in here. So again, we borrowed it from something and try to parameterize to reproduce the patterns. Right? It's actually a question, where is it coming from mechanically? And I probably won't have time to go, but we are working right now on trying to derive these things from the first principles. But yeah, this is what we see in the mechanistic model. This is basically phenomenological, and we pretty much get the same type of idea with a factor of 1,000 speed up. So now we can easily model the number of cells that we need for this type of thing. So the next question is really, how do they know to bias the reversal frequency? What makes it going? And for that, there are 2 types of behavior. One of them is they may produce some chemical, and then they sense the gradient of this chemical by taking temporal derivatives, measure it at the beginning or end of the run, and see if it gets better or worse, and prolong the time in there. Or they can count the contacts and number of cells that go with them and without them. And the reason I think the content of contact was my model of choice is because this is actually what makes them self-organize in those traveling waves. But the way you can get the waves, and it's something very simple, like you can get a student to simulate in the class of the cells. There is basic contact signal besides the sound contact that simulates the reversals. And then pretty much there is a refractory period. Once you reverse, you won't reverse again for some time. After that time, you start counting the contact and see what happens. And basically those 2 things is enough to, in the aligned populations, to them to self-organize into those waves, because what happens is this waves goes and it collides with one another, and they sort of synchronize on collision, and they seem like they pass through one another, but indeed those wave crests reverse from one another. So let me play you a movie if we can get there again. Yeah. So the waves seem to like they're passing through another, but actually the cells that compose the waves just go back and forth, and they reflect from one another in there. And we basically did the tracking, and we did the models and stuff. So that's how the waves come by. So I was a big fan of contact signal. So we basically tried to come up with that and parameterize it to match that bias data. And the short story, it didn't work. You can't get it to work with the contact signal, and the idea is that when you only count in the contacts, the signal is not sort of persistent in space and time. You take a signal with you, it's kind of in this, goes with you, and that's not enough to average a noise. But if it's chemical that you produce, and this is a model of chemotaxis, sort of a standard model of chemotaxis that you can rule, then if there is a cell density fluctuations and there is a fluctuation in this chemical and there's a gradient for it, this gradient will sort of self-amplify and works as some time average thing to reduce the signal to noise in here. And basically what you can see here, contact signal, nothing has happened. Chemotaxis, you start getting streams, and then you start getting the aggregates in the points of intersections of the streams and stuff. So that's pretty much what happens, and indeed, this is sort of an aggregation over a long time. I kind of play the movie in there, but you can see intersection of streams. In the point of intersections, the aggregates will be appearing, and that's sort of what is happening. And again, you get this sort of alignment trend to the aggregate. So you can see that right at the aggregate boundary, the angle is 0. So right around the aggregate boundary, most of the cells move what is that, tangentially to the aggregate, and as you go towards about 40 micrometers, so about 5 to 10 cell lengths away, then it's pretty much plus minus 30 degrees. So that's what this sort of order parameter represents in there. Yeah, each cell produces a signal molecule, and that signal molecule, the time derivative of the signal molecule determines the duration of your movement before you stop and change state. Yeah, they're dispersed, but of course you have density fluctuations. Emergent and low density fluctuation will result in signal fluctuations, and this is self-reinforcing. Not, I mean, they diffuse. There's diffusion. There's no flow. That's a big question. What those molecules are, and we have some candidates. We know that but we don't really know the biochemical basis of that still. We're working with experimenters to figure it out. I think this is something that I would love to get, and this is what we'll get in, but we've been trying to do. So yeah, anyway, so what is it time-wise? So we are almost done, so I'm not going to go too much in this story, but I'm going to be talking to applied mathematicians elsewhere, so I put that in. This is, again, current work with collaboration with some mathematicians there, and what we've been trying to understand is sort of first principle derivation of those sort of active matter type of things, interactions. So, for example, how we can get Ricci-like models from the first principles. And the idea there, so from the sort of binary type of interactions, and we sort of start with some agent-based models with pairwise binary interactions, where Sal basically changes its angle with some delta, small delta parameter, and phi is just a torque that in here as a function of there, some arbitrary function, usually sine of 2 theta. So we start with this, we sort of derive the resulting model, and then we sort of get like a mean field, mean field, and what would be the really an interesting thing that when you start with something like that, and it's sort of similar where some people are doing it, you get additional geometric factor, this absolute value of sine of theta minus theta 1. And this is kind of absent in those type of models. And this is really simple geometric idea. When cells are almost parallel, they have lower probability of collision. So, and as a result, they are less likely to collide and less likely to align in there. And this is something that was sort of missing from many things. Then we can see what it can do. So, for example, we can put some noise and see how sort of uniform distribution of orientation get to partially aligned one. And this is sort of a standard liquid crystal model with just a sine to theta. This is with additional geometric factor. And you can see this Boltzmann model, it needs more signal to overcome the noise in here. Yes, in this case, yeah, it's pneumatic. So it's the function is pi periodic, not 2 pi periodic in there. Mm-hmm. Yep. So it's a pneumatic version of Vishik models and stuff. Yes, Vishik is polar. Yeah, and then what really happened in the true bacterial case is this asymmetric things when cells can either go this way or the other way. But what happens, only one cell changes its orientation. So it's different from small delta in both of them. And when you try to do, you can do also Boltzmann-like derivation of there. And you can get some sort of equation with some interesting math in there. The problem is that, but that's why I'm not going to go in that, when you start comparing this model with agent-based, it fails miserably because the Boltzmann assumption, you have this 2-particle distribution function, you just make it as a product of 2 ones. Because when 2 gas particles collide, they reflect from one another and they never see each other again in the dilute limit. When 2 bacteria collide, they keep moving together forever. And that's what makes those Boltzmann type of models fail. So we need 2 different things, and that's what we are working on now. But again, we were not the first to do the Boltzmann type of distributions, but we were the first one maybe to compare it with the agent, and we see that in this type of situation, even if you put a lot of diffusion, a lot of noise, it's still very hard to rescue this type of model. So, I mean, again, interesting. So we have some additional geometric factor, but we also see that in the sort of this binary collision model, you really need to have high order correlation and stuff. And actually perfectly correlated is better approximation than, or particularly anti-correlated is better approximation than non-correlated in here. So anyways, I'm not gonna be going there. The conclusions are what I show you. I show you this data-driven things to uncover the important correlation of behavior, and then sort of mechanistic, more physics-based things to try to understand which could be the potential mechanisms. And of course, the models, the most informative here are the models that fail. So once we fail, we know we have to go to the drawing board. Now we sort of know what works, but we need to go sort of further challenge it with more data to try to understand some nuances of that in there. But yeah, so that's what happens. And people who did this work, Chris was actually an experimental student who did a lot of data and then spent some time in my lab doing these data-driven models. Rajesh and Zhaoyan did this alignment thing. Patrick is doing this with collaboration with math guy from your age, the self-organization and sort of Boltzmann models of alignment. But yeah, thanks a lot, and I'll be happy to take more questions. It's not about non-binary. No, I'm saying even in the binary limit, even if they're dilute enough to have a non-binary limit, so what is saying, collision probability decreases as cells get more and more aligned. And again, I haven't seen it anywhere. If you've seen it somewhere, let me know. I mean, but we've been very careful. So you have this additional geometric factor of absolute value of sine and theta. It's just the area of space where collision is possible when 2 cells move in. It gets smaller and smaller when the rotations are getting there. So as a result, you get less alignment when cells are almost aligned, and that makes it harder to get a perfect pneumatic alignment, for example. Mm-hmm. Okay, so the beauty of the first part of Datadriven, there is no parameters there. So it's kind of like non-parametric curve fitting, like smoothing or something like that. When all the parameters comes from the sampling there. So, and in that regard, we can see the distribution because, say, for each correlation, there is a spread, and both of them come into picture there. And that's why I'm a big fan of starting with this in there. And then when we do, of course, we need to put some noise in distribution. We try to get more noise because the question always is this signal enough to be an instability to break the symmetry and form those aggregates? Is given the presence of the signal and presence of noise, because cells behavior are very noisy, they're not perfect, and there's cell-to-cell variability, there's time variability, whether that signal is big enough to the noise to get enough of the driving force there. And that's why it's very, very important. And when we do the more mechanistic model, we always try to get the realistic estimates of the noise. Yeah, definitely. Mm-hmm. So when you have mechanistic models, different number for different complexities, right? We had this. I mean, I would say for simpler model, it's probably a handful, like I mean, as I say, speed, reversal frequency, distributions, or the noise terms in there, and then maybe strength of the alignment and something like that. So maybe 5 or 6 for more complicated is there. But I think the idea is that there are some free parameters and there's parameters that we have measurements on. So the number of sort of emergent parameters is usually always a handful. But there are other parameters that we get from data, and we use that to parameterize the thing. So on this type of scale, theory of everything to theory of something, bacteria. But even bacteria, I mean, this is specific for this particular bacteria. But for example, similar type of things, like say, the collective alignment, definitely this happens in other bacteria, like Pseudomonas and stuff. So I can tell you like one example. So again, I usually do problems where I have experimental collaborators because I find the feedback to be most rewarding type of thing. But sometimes things happen. So for example, right when we were doing this collective alignment and saying that reversals kill the collective alignment, there was a postdoc in our center trying to understand sort of immunotherapy for cancer. And what he found is that there is the immune cells that want to come and kill cancer, they can't get through because the fibroblasts on the surface of cancer, they kind of form this barrier and don't allow them through. And he was trying to understand how that sort of barrier is coming from and the alignment of fibroblasts in there. And fibroblasts kind of also long and thin, and they can also kind of go back and forth because the polarity kind of randomly fluctuates. So we've been trying. We've been trying to take our model and apply it for this one. I mean, the numbers are completely different. The scales are different, but I think the physics is somewhat the same. And the same, we see that it's just simply from local alignment, you can get this pneumatic order even locally, not saying globally. So there must be some orientation memory slime trails. There's no slime trails in the fibroblast, but what they do, they pull on the polymer network around this. It's like a extracellular matrix or something like that. And when you pull on the polymer matrix, it aligns. And this alignment helps other cells to align. So there's still orientation memory. So I can say that there are other systems quite parametrically, but the same physics can apply. Yeah. So they're nearby that we don't that haven't been studied much. but I don't think there's nearby that does the same thing with different things. But what we really try to do is the mutants of this bacteria. You can delete this, delete that, and patterns change. So we are trying to form this loop. This is something that we are doing right now. Or which? Yeah. Oh, that's super important for their cycle. This is like end of their life cycle. I mean, I didn't talk much about that, but as I said, they're very social. They need others, and when they are starved, what they do? They kind of forms those spores. It's like kind of like a seed for a plant, which has all the genetic information, but it waits for better life to come. And when you and many cells do the sporulation, but they usually do it in place. But those cells come together, form those agglomerates, like you call aggregates, and they form it inside of them. And they are like almost microscopic. You can see them with the naked eye. They can be 0.5 millimeter or 30 millimeter across sometimes. And that one first allows like a bird or something to get stuck to something and carry it elsewhere. Or it's when the conditions improve locally, they all together, and there are hundreds of thousands of cells in that agglomerate, and they all germinate at the same time, and they all become back to their original cell. So this is why why it's so important for them to come together and do together. There are some other things that when they do that, some cells will not make those spores, but what they do, they will produce sugars like crazy, and they will make a coating of that aggregate, which will be sticky, so allows it to carry, and will be a little bit protective. Yeah, so there is some like, there's definitely evolutionary advantage. There are some cells that sacrifice themselves for the sake of community. So there is a very interesting evolutionary ecology aspect of that, which, again, I didn't talk about much. All right. Yeah, I mean, I'll be happy to stick around and talk to people offline more if interested. Okay.